So it's really an honor to be here today. I'm very glad. So today I will talk about the research that I have been doing recently at the Department of Biological Psychology at the Free University in Amsterdam. But first, I've got to thank the organizing committee for inviting me and coordinating this visit. I'm really very proud of you, and you did a very good job. So let's start today. I will start with the genetics of fertility and twinning. This was one of the projects that I was working on since I moved to Netherlands. So I did my bachelor's degree at the University of Tunis. I started a doctoral degree at the University of Ivry-Van-Disson, where I studied the genetics of rheumatoid arthritis, and actually Dr. Sani and Dr. Reem were present at the defense of my PhD in 2009. Then I started my first postdoc at the University of Tokyo in Japan, where I studied the genetics of cancer and complex diseases. It was a great experience. I strongly advise you, if you have any opportunity to visit Asia, please do so. Then in 2012 I moved to Netherlands, and I started as a postdoc at the Free University. My first project was the genetics of addiction. And then after a few months, since I worked at the Netherlands Twin Register, so I started to study twinning and fertility. So here the layout of my presentation, so a bit of background. The Twinning Genetics Consortium I initiated with my supervisor at the time. The first results were the meta-analysis model of these twins, and then the last point is about the importance of collaboration in science, and an example of the UK Biobank, and another consortium which called human reproductive behavior. So the Netherlands New Register was founded on February in 1987 at the Vrije University in Amsterdam, and there is one student here from Vrije University, with the purpose to conduct scientific research. A large number of families with young twins are registered with the NTR. These twins are followed from birth into adulthood. The aim of the NTR is to examine the contribution of hereditary predisposition to personality, traits, to growth, to development, to disease, and to risk factors for disease. The NTR followed twins and their families longitudinally by means of questionnaires, surveys, lab visit, home visit, and emails, and so on. Because it's important to follow these people longitudinally. As you may know, at many points in the span of a lifetime, the unique biology of the individual will play an increasing role. The figure of the slide shows the timeline of sequencing applications in medicine from pre-warm to tomb. We can now apply medical genomics at many points during an individual lifespan. For example, prior to conception, a couple can have a genomic screening for important recessive alleles. An expectant mother at 8 to 12 weeks of pregnancy can now have a single tube of blood used to accurately assess chromosomal abnormalities of the fetus, determine the gender, and even whole genome sequencing of the fetus is performed. At birth, sequencing the genome of the newborn can be used to rapidly diagnose many conditions. Ultimately, omics information at young age will be useful by providing susceptibility to various medical conditions that have actionable prevention strategies. So, for example, at the end, when a sudden death happens, molecular autopsy can help and diagnose the cause of death and can prevent from the death of other family members. At NTR, which is unique worldwide resources of twins and their families, we use twins to study at which extent the contribution of DNA variants to differences between humans. Multiples are not different from singletons, but the fact that with the help of twins we can determine to what extent differences are contributed to hereditary and environmental is unique. For example, one person has high blood pressure and the other low blood pressure. Are these differences the results of environmental factors such as the use of salt? Or are they hereditary? Usually both factors are very important And the aim of the research is to determine how large is the contribution of genetic factors. For further readings, I suggest to read these two papers written by my colleagues at the department to know more about twin designs and twin studies in the omics era. They are in Nature Review Genetics. So these slides represent the data that we have at NTR. So that's from December 2017, we had a total of 220K participants in the register. They are twins, multiples, they are sibs, parents, and offspring of twins and offspring of sibs. We also include the teachers of the twins because we did studies on whether it is beneficial for twins to stay in one class or to separate the twins for identical and heterozygotic twins. Some of the data we have at NTR, so it's very rich data. We have, as I said, surveys, we collect DNA, we have genotyping, whole genome genotyping, we have DNA methylation, RNA expression, sequencing, metabolomics, microbiome, and so on. So this data is well structured into different databases. The Panther database is the first. So the Panther database is where we collect the information about the addresses, the names of the participants, and then we connect this database to phenotype databases, where we collect data, phenotypic data surveys, and we assign a different number for each database to avoid, to keep privacy of the participants. Then, for some of the participants, we have genotype data, and such as DNA genotype, sequence, gene expression, epigenetics, and we have unique identifiers for the genotype data. So because we have a lot of data and we have a whole genome high throughput data, we cannot analyze this data on a single computer, so we use the facilities, national facilities, high performance computing and data infrastructure, which I think every country should have this kind of infrastructure to study high throughput genetic data at the population level. Here is another paper about the implementation of a twin register for studying behavior and genetics. How do we determine whether hereditary predisposition plays a role in behavior and disease? To do this, we need twins, both monozygotic and heterozygotic twins. Monozygotic twins, as you may know, are genetic copies. They are 100% identical. In contrast, heterozygotic twins are genetically similar to ordinary brothers and sisters. They share on average 50% of the genetic material. Twinning. Twinning, this fascinating trait, has some increased risk for the mothers and offspring, like preeclampsia for the mothers and stillbirth for neonatal, late and prenatal birth. However, it's a very good measure of fertility, so both at the individual and at the population level. We use this ratio of DZ-MZ twinning as a marker of high fertility, which reflects the frequency of double population fertilization of both open and the maintenance of multiple pregnancy. For monozygotic twins, there are no clear evidence for the application of genetic factors. However, for DZ twinning, it's a complex trait. The tendency to conceive spontaneous dizygotic twinning is a complex trait with important contribution from both environmental and genetic disposition. Significantly higher multiple birth rates have been reported in mothers who, for example, smoke, who have high BMI, and who are tolerant. And the previous study showed that, family study showed that high frequency of DZ twins existing in females of a relative of mother of DZ twins. Like if there are a woman who has twins, her sister has higher chance to have also twins. So the the chance, I don't like to of the risk, but the chance of giving birth to these twins is about twice the population average in these families. Another factor is the maternal age, which is an important chance factor, which increases with age. For example, it increases fourfold between the age of 15 to 37 years. This was very old research by one of the pioneers in this field, Belmar. What we know about the genetics of this twinning? As with candidate gene studies in most complex traits, the results have been mixed. Many of these studies were also statistically underpowered. So, one study by Parvin and al. In 2006 identified the association of rare variants in one of the growth differentiation factor 9, GTS9. Another study, a large linkage analysis in 523 families, which is about more than 1,000 mothers of these twins, produced a max load of 2.7 on chromosome 2, where it is located the follicle-stimulating hormone receptor. Why we want to study twinning? Knowledge of the twinning genes may identify key mechanisms controlling ovarian function and can provide a greater understanding of female fertility and infertility, which is the most common reproductive disorder. So, as I said in the beginning, I joined the department in January, and in November we initiated the Twin Genetics Consortium together with my supervisor, Professor Dorit Boomsma. So at that time we collect about 6 twin registers, so they are working on, they have data on twins, and we initiate this consortium together from Finland, Sweden, UK, US and Australia. 5 years later, in 2017, we have more twin registers in this consortium. We have about 10. So, because it is important to study as many as possible as twins to increase the sample size and to be able to find the genes. What we are studying in this consortium. So, the first trait is being the desert twin. Here, the cases are defined as desert twins, and the contours non-desert twins. They can be mothers and fathers of answer twins, because in the twin register we have mostly twins data and their families. But they can also mother and fathers of singletons or occasions or healthy controls from the population. So we expanded the criteria of controls because we wanted to have as much samples as possible. So because of we can include other data that are not from twin registers. The second trait is being the mother of these twins. The cases here are mothers of these twins with documented history of spontaneous twinning. We try to focus only on spontaneous degenerative twinning, not IVF, and the controls can be non-mother of these twins. So mother and father of MZ, or singletons, or as the previous case or healthy controls. The third trait is being MZ twins. As I said, it's not genetics. At least so far, there is no evidence that identical twins are genetic. So, but we are studying this trait, and there is some previous studies showing that there is some segregation in families, like in Germany. They found families with many pairs of MZ twins. So here the cohorts that are involved in the first study, mother of DZ twins. We have a cohort from Australia with about 600 cases and more than 6,000 controls. A cohort from USA, 500 cases and nearly 2,000 controls, and our own cohort with 800 cases and more than 4,000 controls. So in total you don't have a large sample size, you have about 2,000 cases and 13,000 controls. A very important consideration is that we selected our cases not to be conceived, the twins not to be conceived after IVF. So we excluded all the mothers, but they reported they have been using any kind of fertility treatment, like hormone stimulation or IVF. We we avoid this, we excluded the samples. For the controls, we tried to do our best to use only controls that didn't have any relationship with twins. But this was not possible for all cohorts, but we tried our best to avoid parents with diesel twins, to avoid any bias in the study. So very quickly on the quality control steps for each cohort. So we wrote SOP, standard operation procedure, where we include all the steps, all the inclusion and exclusion criteria we want to study, and the analysis, the association analysis that we want to perform. And we asked every study to send us their summary statistics, and we do the meta-analysis, the QC and the meta-analysis at a centralized place. So these are some of the criteria. We used also correlates. We adjusted for sex and principal component for the population stratification. On the right side of the slide are the QQ plot. I'm not sure if you're familiar with it, but quickly. The QQ plot shows the expected distribution of association test statistics on the x-axis across a million SNPs compared to the observed value on the y-axis. Any deviation from the x-y lines implies a consistent difference between cases and controls across the whole genome. So a clean QQ plot, like the one that I was showing on the slides, should show a solid line matching x and y until it sharply curves at the end of the time. So here there is no deviation, so that means that there is no population stratification, there is no affiliation, but it shows also that there is no signals at the individual cohorts. So this is why we choose to do a meta-analysis, and here the study design. So we combined each summary statistics of the 3 cohorts, and we ran a fixed meta-analysis, fixed effect meta-analysis based on betas and standard errors. We selected the top independent SNPs, and then we performed an independent replication in different population. In this case, it's Iceland population. It's from DCO. It's quite a large sample size, 3,597, and the number of diesel and nearly 300K controls. This means that shows p-value below 0.05 are selected for a combined analysis between the discovery stage and the replication stage. Here are the results. This slide shows a Manhattan plot. On the y-axis, you have the strength of the association between the cases and controls, which is shown here as minus log 10 p-value. And on the x-axis is each point represents a position in the genome, a single nucleotide polymorphism. There are 2 lines. The blue lines represent the suggested p-value, which is 110 to minus 5, and the red line represents the genome-wide significant p-value after morphology correction. So any SNPs above the red line is considered as genome-wide significant and worth follow-up replication and validation. So as you can see here, we found 3 hits. The first hit is on chromosome 1. It's an intergenic SNP between angiotensin gene and a gene involved in glycosylation. The second hit is quite known candidate genes for twinning, which is the follicle-stimulating hormones, beta-subunit, which is in conjunction with LH, induces egg and sperm production. So it's a very good candidate and very nice that we picked up this gene. The third gene, however, is located on chromosome 15. It's called SMAD3, and it is a transcription modulator activated by the TGF-beta pathway. So it is thought to play a role in the regulation of carcinogenesis, and there is no previous study implicating this gene in twinning or fertility. So here are the results in terms of numbers. So the first SNP is on chromosome 1. Unfortunately, the SNP did not replicate in the recorded population, although it showed low allele frequency, RAF for the risk allele frequency, and the 2 other SNPs were replicated in the decode population. We also did some functional analysis. We studied the FSH level in these mothers of twins, and we found that FSH level is associated with the toxins in FSHD with a p-value 2.3 10 to minus 10. So each risk allele giving an increase in FSH level of about 1.11 SD units. So we tried to link twinning to fertility, and we looked at the association of these twinning SNPs in other fertility-related measures. So here are the results. Sorry for this complicated slides. So we showed that FSHD variant is associated with an increased risk of twinning. It is associated with an increased FSH level. It is associated with early age at menarche in terms of years, early age at menopause. It is not significant with the traits having children. It is significant with the number of children, and it is associated with early age at first child, and it shows a trend of association with age at last child. It's very important, the last line, it is associated with PCOS, polycystic ovary syndrome. So the higher chance you have that means the lower chance you have PCOS, which is logic. The second SNP, however, is associated in SMART3 is associated only with later age at last child. It's not significant with other measures of fertility. The data showed in this table are coming from our own GWAS, from the default population, from papers published by colleagues in the UK, and from the default population. So this slide summarizes our results. So we concluded that the genetic variant in FSHD is responsible for higher secretion of FSH, which in turn will lead to more maturating AIDS and multiple population. This increases higher likelihood of either twins, and it is also associated with other fertility traits. On the other hand, the genetic variants in SMART3, we thought our hypothesis is that is involved in the regulation of the response of the ovary to FSH. So understanding the role of SMART3 will offer another opportunity to optimize the responsiveness and minimize the risk among ART recipients, IVF-OM. These are the implications that we are studying currently to know more about the function of the SNPs and the implication in clinics. So we did also some risk predictions, and we found that when both variants are present, a woman has a 29 greater chance of having these twins if she had both risk alleles. Recently, the FSHD variant has been also associated with a shorter menstrual cycle, and also in endometriosis. So there are more and more roles of these variants in twinning and fertility. So this led to the publication in 2016 in American Journals of Human Genetics, which and a follow-up study, follow-up paper, where we addressed the question how to present the results in terms of effect size in a manner that is accessible and comprehensible to the general audience. So we avoided the standard effect size, such as odds ratio and relative risk, as this requires some knowledge of probability theory. So we rather convey these results in terms of the conditional probabilities expressed in natural language. So I invite you to read these two papers if you are interested. So the paper has been selected in 2016 as the best, among the best papers in American journals for groundbreaking research published in late 2015 and early 16. It has been also worldwide covered by the media. Many media around the world covered these findings of the twinning genes, and here are some examples. I'm particularly proud of the science blog, which reported on the paper, because our first submission was the science, but they asked us to increase the sample size because, let me come back to the Manhattan plot. So I talked about this blue line here, and the signals that you see between the blue line and the red line are all suggestive SNPs that they may have. If we increase our sample size, we may find more genes. And this was one of the comments from the science paper. They say, wait and increase your sample size, you will find more genes. So at that time, we would like to publish as soon as possible. So that's why we decided to publish in American Journal. But nevertheless, they picked it up in the news and they wrote about it. So there is actually a way to know if your research is appreciated in the media, who is talking about your research. So this is a tool called iMetric. It does this by pulling in data from three main sources: social media like Twitter, Facebook, Google+, Pinterest, and blogs, and also traditional media like both mainstream, the big journals, Guardian, New York, and also specific scientific journals. So, and here, as you can see in the slides, it has quite high scores, and it is scored number 4 among the output of the American Journals of Human Genetics. So this up-to-date data. Since these tools were used, this paper was ranked number 4 in all the output of American Journal. So we did not stop there. We continued our recruitment for cohorts who have twins and genome-wide data. So just this slide is to mention that we have been working on increasing the sample size, and for example, for the being mother of these twins, we now doubled the sample size of the cases. And we also collected data on being MZ and being DZ, but today I will focus only on DZ twinning. So the other takes are its ongoing work. So what are the results of the new meta-analysis of mother of DZ twins with more than 3,500 cases, so almost double the sample size of the first paper. And here are the results. So again, a Manhattan plot, and as you can see here, we validated our first hit, so that's good. It's not false positive. We found also the same hit on SMART3, but you can see here there is other genes, other hits that starts to pop up. The hit on chromosome 2 is located in the FSH receptor. There is another gene here which is involved in ovulation, in the maturation of the eggs. There is another candidate gene on chromosome 12 and another hit on chromosome 16. So you see, with increasing sample size, we start to see some clear hits popping up. So this was at the SNP level. So to have more power and to know more about this association, we did a gene-based test. So instead of looking at the single SNP, we looked at the SNPs that are within the gene. And here, the genes I mentioned before, some of them are genome-wide significant, like the genes MTFR1, which is involved in mitochondrial function and highly expressed in human metaphase 2 oocytes. And the expression of the gene is significantly reduced by age. The other genes in INPO8 is required for the TGF-beta activated SMAD2 and SMAD3 to translocate into the nucleus. So you see that they are all in this TGF-beta, so that made a lot of sense. So in addition to finding more these genes, we are also, in the meantime, we are trying to link this genetic data to clinical data. We are studying the effect of FSHD SNPs in preterm birth among the mother of twins. And we are recruiting also non-Caucasian ancestry because the GWAS and the meta-analysis I showed you were based only on European ancestry. And because we know also that Asians have lower the rate of twinning in contrast to Africa where they have a higher rate of twinning. So we want to see what are the mechanisms involved in this, so why there is this discrepancy in the twinning rate around the world. And it would be good also to study the Tunisian population. This is a question I had always from my supervisor, whether we can study Tunisian twins. We are also working on whole genome sequencing of a large pedigree, which contains 11 mothers of lizard twins. There are 22 pairs of twins in this pedigree, and we first did the linkage analysis and IBD mapping to select the regions, to select the best samples or best informative samples, and we see the arrows in the red show the 4 samples that we sequenced whole genome at 35x. So this is a work from my student in the USA. So I mentioned about UK Biobank. We are also trying to collaborate, or we collaborated already with UK Biobank. It's a large biobank of about 500K participants, whole genome genotyped. And they made these resources available for the researcher around the world. So we applied to have access to this register and to try. This is the paper about UK Biobank. They used a chip, genomide AFI, with these characteristics. So they coupled the previous genomide significant hits for many diseases. And they used also a known coding variation, and so these are the things that are included in the chip. So we have been looking at the data in this UK Biobank. If there are any twinning data, we found one question, which is, are you part of multiple birth? Are you twin, triplet, or other multiple birth? The question was asked if yes or no. So you see here about 11,000 said yes, I am part of multiple births with a twin, but we don't know whether it is diesel twin or whether it is MZ twins. And on the right picture, it's a distribution of age of this sample. So we ran GWAS on being part of multiple births in this data. So the final numbers are here after quality control. And here are the results. So again, magnetic dot, and you see here 2 hits, one in chromosome 11, which corresponds to the FSHD, and another one on chromosome 15, which is near SMAD3. So this is very good validation of our previous results because it is an independent population, and we found the same hits. And to notice that this is not the same trait as the first of our paper, because here we are looking at the twins themselves. So it's a proxy of our phenotype. But it's good that we found this hit. So, but we did not stop there. So because we don't know if they are MZ or DZ, so we want to refine the phenotype. So what I did is I excluded all the twins that were born after 1970. This is 1969 was the year where the clomiphene was introduced in the UK. So to avoid any bias introduced by fertility treatment, we removed the twins that were born after 70. And we also, using the matrix, the kinship matrix, which is a resemblance between, genetic resemblance between individuals, we removed the MZ twins from the study. So there are about 200 twins, MZ twins we removed. So now we ended up with 8,962 cases, twins. These are twins or higher multiple, but they are, since they are there, so we can safely say that this is these are twins. And 400, more than 400K controls. But for the controls, we don't know if they are relative of twins, so we don't have control over that. Interestingly, the hits or FSHV is still there, and the hit in SMART3 or near SMART3 is getting stronger and it's becoming genome-wide significant. So this is a very nice example of the refinement of the phenotype. So we shouldn't worry a lot about increasing sample size. We should very carefully choose our phenotype for such kind of association and to avoid any compound. So here are the regional association plot of the two hits. So here are the hits on chromosome 15. You see this is intergenic between PS1 gene, which is involved in testicular germ cell carcinoma, and SMART3 is not is here, but the association is mainly between these two genes. And here additional association plot of chromosome 11. So you see various signals here surrounding in SHD. Since UK Biobank, they had a lot of phenotypes, a lot of traits. We studied also the genetic correlation between twinning or multiple birth and these traits, and these are the significant traits. So interestingly, you can see that it is positively correlated with weight, with body size, with BMI, which we know already from epidemiological studies. It is inversely correlated with the age of death of the father, the age of last birth, and the age of menarche, which we already showed in the paper. So this is a very good validation. This strong correlation here with TR doctor is suspicious. We don't know what it is exactly, but TR doctor is the time. How many times did you visit the doctor when you were a child? So it may indicate that twins, they have a very good care when they are young. So, but we are looking into that at the moment. We are also collaborating with a group from Oxford. They are working on reproductive behavior. It's a consortium called Human Reproductive Behavior. And the aim of the study is to identify genetic loci that are related to the age at first birth and the number of children ever born, and to examine their biological function. Is there any function behind the number of children that we want to have and the age where we want to have the children? So there are 250K individuals participating in the age at first birth, and more than 300,000 individuals in the number of children ever born. So this is the result of a meta-analysis. There are 62 cohorts participating in this analysis. And the question is, as I said, how many children do you have and at what age you have your first child? So these are the results of the meta-analysis. We identified 12 independent loci, 10 which were not previously anticipated to influence reproductive behavior, that were significantly associated with age at first birth and number of children. So we encountered some very exciting biological follow-up findings. Some of the lead SNPs are related to critical fertility process, such as follicle-stimulating hormones. So we found the same gene that was implicated in twinning. We found it significantly associated in this GWAS, estrogen, growth in ovaries, spermatic differentiation, development and diseases like endometriosis and PCOS. So here I want to switch and to talk a bit about the use of this genetic variants associated with twinning and fertility in general. This is a company in the USA based in New York, which is called Sematics. They are using the data that are published by us and other groups working on fertility around the world to try to make a genetic test for women who are trying to conceive and to tell them what are their chance to have babies. So it's based purely on the scientific data published by a group like us. So this is an introduction of this kit. It's called Fertility. So this is another example of a company, direct-to-consumer company. It's a genomics company based in Silicon Valley in California. It's called 23andMe. It's a private personal genomics and biotechnology company. It is a saliva-based direct-to-consumer personal genome test. Here, they are not interested particularly in fertility, but they are interested in ancestry and other disease and traits. So there is one DNA test available which genotype around 570,000 SNPs of the human genome with Illumina chip, and they provide ancestry and health information for their participants. So far they genotyped about 2 million participants. It's a huge effort. They were, the data that I showed in my paper were partly also from 23andMe, and it seems they were interested in twinning, but I have been trying since 3 years to include their data in our meta-analysis, and every time they say we don't have enough twins. So, but at least they are making some efforts, and this is a Twitter from 23andMe where they asked to recruit MZ and DZ twins. But the last time we had an answer from 23andMe, they said that they are willing to replicate our findings. So it's a good sign. So for the sake of time, I will skip this 2 slides. Yeah, I want to finish here by thanking my colleagues at the NTR and Avira department and lab. New in Amsterdam and Stuttgart, USA, and all the twinning genetics consortium members from different countries. And my last slide is just advertise about a new research master. For maybe you are not at that stage, but if you know someone who is interested in twinning and genetics research, this year we are starting a new research master called Genes in Behavior and Health. And during the summer, I'm one of the organizers of the course on nature and nurture twin research and human genetics. It's a 2 weeks course, and if you know someone who is interested, you can approach me. That's it. Thank you for your attention.
In science, there are trends. So some topics, are very in for a certain period of time and then they die out. And I think twins have been in forever. They have been in, from finding the risk of, breast cancer to recently with the science and space research about twins. So what we've been doing is very, very interesting. Now and this is the thing I looked at your study was the cases that you're looking at, or the population that you're looking at, which is mostly Caucasian. Then you hinted on adding more from Asia and from Africa, especially from Tunisia. So I was curious how you're going to do that. Is it just collecting data and bye-bye, or how are you going to do that? Thank you.
Thanks for your question. So what I hope really is to establish a Tunisian twin register here, because not with the aim to study twins and fertility, but as you have seen in the slides of the Netherlands twin register, we are studying a lot of traits and diseases, and we can establish the heritability part of these traits and diseases in the Dutch population. So if I'm quite confident that there are the brains to do this in Tunisia, but we need to, there is a need for some, how to say, volonté scientifique, mais la volonté politique de faire ce genre de choses. So I hope with collaboration with Sonia and Reem and Alia, who presented last day. So we need to collect samples on Tunisian samples and try to seek fundings from international agencies and do the research in Tunisia and send the samples overseas. So we can do that. It's not a big deal, but just we need to have the tools for that. Like an infrastructure for computing, high performance computing is really needed. So because this is high throughput data, and if you have NGS data, you cannot analyze this data on your single computer. You need to have a server, you need to have a cluster to do this. And all the data that I'm analyzing are based on or done at the national server. Yes, Sonia.
Thank you. Thank you.
Okay.
Thank you for the nice presentation. I have just a naive question. You mentioned correlation between 3 genes and 20, and I didn't understand how you validate experimentally this correlation.
Yes, so we did already validation by replicating these associations in an independent population. It's already validation. Now we are doing the experimental validation by looking at the genotypes of this woman and how they are responding to IVF. So knowing the SMART3 variants, we can, for example, tune the hormone stimulation. So one of the risks of the hormonal stimulation is the hyperstimulation. So if we don't know if the woman will highly respond to hormonal stimulation, so it may lead to hyperstimulation syndrome. So by knowing her genotype, her SMART3 genotype, and FSH and other genes, we can tune this stimulation. So there is a lot of applications in cleaning for these genes. Yeah, sorry.
Thank you. Thank you very much for this impressive work. It's really great. I'd like to make a comment because several among the young people who are present here, they would say that you had the chance to have had your studies abroad. And yesterday we discussed about chance and the chance to get a job or chance to be where you are. And I want to say that la chance est soumise aux esprits préparés. Chance smiles to prepared minds. What I like to do is to propose, because yesterday we had that very nice session about breakthrough and how to make it happen. I'm sure that several people here would like to do the same work as you presented, and even if we are in different conditions. What I'd like to propose is, in the frame of H3 Africa and other projects who are currently being led here in the Institute, in collaboration with the Young Association, the Association of Young Researchers in Institut Pasteur, we make a workshop. And if you could contribute to it by saying what is behind the scene. I mean, besides getting in consortia, there are conditions to get in those consortia. There are conditions in which people are recruited. So if we could set up at least 2 scenarios. One scenario on how it happened abroad with this strong political will, and how it could happen here in Tunisia by the strong young researchers' will. And I'm sure that together we could build something that works. So
of course, and yes, I'm really very keen to help in that matter, because, as you say, it's not easy. So this study is published, so it was taking hard work, really hard work, and my experience in Japan, it was not that easy. So I say now 2 words, but with the time, it was at that time it was really hard. So we were working for more than 12 hours per day. And I was looking forward when I finished the project to come back to Europe or to Tunisia. So it's not easy. You need to work very hard, and so you need to be patient. But I'm sure you have the skills to do that, and I'm willing very much to help. That's why I'm here today, so I'm open to any suggestion, to any collaborations, because I know what it is, I know the conditions that are working in, so yeah, very good initiative, Sonia, and please let me know what I can help.
Okay, thank you, Hamdi, for this nice talk. I would like to ask if there's any rare variants that have been identified as associated with twinning using, if you like, whole genome sequencing or whole genome sequencing. I mean, variants with a higher order issues.
Yes.
Yeah.
There was a study in 2006, I think, which I showed where they found rare variants in GDF9, but this was not replicated. And now,
I'm asking for recent studies with.
yes, we are looking into this large pedigree. So this was one of the largest pedigree in Netherlands in register, and we are. Where is it? Here, yeah. So with Jeff Beck and the student, PhD students in USA, we sequenced already these 4 samples, and now we are looking at the rare variants with GATK. So there is some good candidates, and we are digging into that. I hope we publish. So I will send you the paper.
Okay, just one other question for the polygenetic score.
Yes.
You calculated with the 2 variants that you identified, or with other variants?
Yeah, we did a polygenetic score based on the whole genome, and we found, so we predicted the twinning in Icelandic population. So we used the whole genome.
Pragmatic studies that have been done on the variants that have been identified, like to find total variants or with functional analysis for those that you have already identified on SMAD3 and
yeah, so
computation like imputed variants
yes.
that can give higher or stronger signals.
Yes. So with this large pedigree sequencing, we are focusing first on the regions where it's SMAD3 and SHP. So we are looking into this at first priority before looking at other regions or in the genome. So it's very much work in progress.